Yes, and did you go through these pages? Yes, did you read this page? The labeled antibodies are then introduced into the patient's blood. ये पढ़ लिया है?
Yes, miss. Yes, miss.
The position of the labeled maps indicates the position of any blood clots. So now you can see that we are making use of the specificity of the antibodies to help us in diagnose diseases. Because हम जब diagnose करते हैं तो हमें we have to be very specific about some facts कि अच्छा हाँ इसको ये बीमारी है ये करना चाहिए, right? अच्छा, not only the maps are used to diagnose diseases but they are routinely used in blood typing. हम जब अपना blood group मालूम करते हैं तो वो भी maps के थ्रू ही करते हैं. किसी एंटीबॉडी को किसी भी इंसान के ब्लड ग्रुप के एंटीजेंस के साथ मिक्स करते हैं to see the response, right? Okay, there is a question and let me in fact share that question with you. So just a minute, I'll have to stop sharing my screen. I have that question. It is relevant to this topic. One of my students actually shared these questions with me. Okay, there you go. I am sharing my screen. Okay. Look at this question. A person's blood group is determined by antigens present on the red blood cells. जो अभी मैं बात कर रही थी. The table shows the antigens and the antibodies in the blood of people with different blood groups. अब देखो आप, blood group A वाला जो शख्स है, he has the presence of A or B antigens on red blood cells. उसके पास A ही antigens हैं उसके red blood cell के surface पे. Presence of antibodies to A or B in plasma. He has antibodies which are anti-B only. इसका क्या मतलब है first row का? Blood group is A. The antigens that he has on the surface of his red blood cells are A only. और उसकी बॉडी में जो एंटीबॉडीज हैं या कभी अगर बनेंगी भी तो एंटी ए कभी नहीं बनेंगी. एंटी ए इसलिए नहीं बनेंगी कि ए तो वो खुद है ना. A is his own self antigen, so his body will never make anti-A antibodies. ठीक है? अगर उसकी बॉडी ने कभी कोई एंटीबॉडी बनाई भी किसी ब्लड ग्रुप के खिलाफ, so it will be anti-B antibody. अच्छा, फिर एग्जाम्पल किसकी दी हुई है? अ पर्सन हु हैज ब्लड ग्रुप बी, तो उसके पास बी एंटीजन ही होना चाहिए और एंटीबॉडी ए होनी चाहिए, एंटी ए. इसका मतलब अगर हम इसमें ए वाला ब्लड डाल देंगे तो this person will start making anti-A body because his immune system will consider the A antigen as a non-self antigen. ठीक. अब एक तीसरा शख्स है जिसका ब्लड ग्रुप है ए बी. सो ही ए बी का मतलब इसके पास ए एंटीजन भी है बी एंटीजन भी है. तो अब जब ए और बी दोनों एंटीजन हैं तो एंटीबॉडी कोई भी नहीं है, राइट? अच्छा, एंड देन देयर इज अ पर्सन हु इज ब्लड ग्रुप ओ. ओ वाला जो ब्लड ग्रुप है ना, ही नाइदर हैज ए एंटीजन नॉर बी एंटीजन और इसके पास दोनों एंटीबॉडीज हैं. ठीक है. नाउ द क्वेश्चन इज के ड्यूरिंग अ ब्लड ट्रांसफ्यूजन इट इज एसेंशियल that the person who receives the blood does not have antibodies to the donor's blood. ठीक है. Which blood groups can be given to a person with blood group B? Blood group B को हम कौन सा blood दे सकते हैं? G girls, what's the answer? Blood group B को B दे सकते हैं?
Miss, is B the answer?
B the answer. B में B भी है and A भी है. How would I give AB blood to a person who has B? Who is blood group B? अगर वो blood group B है तो मैं उसको A एंटीजन भी दे दूं और B एंटीजन भी. चलो B एंटीजन से तो वो एंटी B एंटीबॉडी नहीं बनाएगा, लेकिन जैसे ही वो A एंटीजन को देखेगा he will start making anti A antibody. तो क्या हो जाएगा? There will be a transfusion reaction and this person will die. कौन सा दे सकते हैं हम? Come on, think. बी तो दे ही सकते हैं, ठीक है, बी बिल्कुल दे सकते हैं. तो उस हिसाब से देन यू हैव टू सिलेक्ट बिटवीन ऑप्शन ए, ऑप्शन बी को हमने हटा दिया, एंड ऑप्शन डी. Yes,
Miss,
Alia?
can it be O?
O, yes, it can be O. Very good. Why can it be O? Because O doesn't have any antigen. O के पास तो कोई एंटीजन ही नहीं है, ठीक है. तो अब आप अपने आप को इमेजिन करो, अपने आप को सोचो, यू आर ब्लड ग्रुप बी. In an emergency situation, you need to be transfused blood, otherwise you will die. So the only two blood groups that can be given to you in an emergency situation is either your own blood group type, which is blood group B, or O. Why O? Because O doesn't have any antigen. तो जब एंटीजन ही नहीं है O के पास, तो B वाला जो आदमी है जिसको आप ब्लड देना चाह रहे हो, वो किसके अगेंस्ट एंटीबॉडी बनाएगा, ठीक है. C नहीं हो सकता क्योंकि C में एंटीजन A है. B भी नहीं हो सकता, B में भी एंटीजन A है. और A, ऑप्शन A भी नहीं हो सकता बिकॉज ऑप्शन A हैज A एंड B. तो वो A के खिलाफ तो एंटी A एंटीबॉडी बनाएगा ना, ठीक है जी. आलिया, आमना, परीशे, हैव यू अंडरस्टूड दिस कॉन्सेप्ट और नॉट?
Yes, miss. Understood, miss.
अच्छा, ठीक है. Okay, there's another question. Acha, we'll do this question afterwards. Let's first try to. So MABs are being routinely used in blood typing before transfusion, tissue typing maybe before transplant. So you need to know where are MABs used. There are now many MABs available to diagnose hundreds of different medical conditions. For example, MABs can be used to locate cancer cells which have proteins in their cell surface membranes that differ from the proteins on normal body cells and can therefore be detected by antibodies. कितनी जबरदस्त चीज है कि by using MABs we can locate कि cancer cells कहाँ हैं. Why? Because cancer cells हमारी body के normal cells की तरह नहीं होते. The antigens that they have on their cell surface membranes are different from the antigens that are found on the cell surface membrane of our normal body cells. तो अगर हमारे पास कोई monoclonal antibody है जो हमने body के बाहर बनाई है through making a hybridoma, we can inject that antibody to a certain person. अगर उसकी body में cancer cells हैं तो वो जाके ठक करके उस antigen पे जो cancer cells के cell surface membrane पे लगे हुए हैं वो जाके antigen antibody complex बना लेंगी. Monoclonal antibodies can be also used to identify the exact strain of a virus or bacterium that is causing the infection. This speeds up the choice of the most appropriate treatment for the patient. For example, let's suppose कि a person is suffering from malaria. ठीक है, अब मैं आपको एक मलेरिया की एग्जांपल देती हूँ जो आपको समझ आएगी when we will be moving on to chapter infections. Now this person is suffering from malaria, right? तो इसका मतलब क्या हुआ कि inside his body, especially flowing inside his red blood cells, let's say ये सारे इसके red blood cells हैं, right? और red blood cell के अंदर plasmodium है. Plasmodium is the pathogenic agent for malaria. ठीक? अच्छा, a plasmodium of course has antigens on its surface. ठीक है? अब patient आ रहा है doctor के पास and the patient has fever but the doctor doesn't know the cause of the fever. So what does the doctor say? Go and get your blood tested for malarial antigen. And when I say malarial antigen, I actually mean the antigen which is found on the surface of the plasmodium. ठीक है, विच इज अ पैरासाइट। तो हम क्या करेंगे, मलेरियल एंटीजन को कैसे डिटेक्ट करें? इफ वी हैव द एंटी मलेरियल एंटीबॉडी या एंटी प्लाज्मोडियम एंटीबॉडी अगर हमारे पास है, हमने बनाई है थ्रू हाइब्रिडोमा टेक्निक, हम इस पेशेंट का ब्लड ले लेते हैं, ड्रॉप ऑफ ब्लड, वी टेक अ ग्लास स्लाइड, देयर इज द ड्रॉप ऑफ ब्लड, और अगर इस ब्लड के अंदर ये मलेरियल एंटीजेंस हैं, राइट, द एंटीजेंस आर देयर, एंड इफ वी हैव द स्पेसिफिक एंटीबॉडी, ये वाली एंटीबॉडीज, we will use these monoclonal antibodies, ये जाके antigen antibody complex बनाएंगी and the reaction will be positive. Always remember, whenever an antigen and antibody, they are complementary to each other, they form an antigen antibody complex formation, और कुछ reaction होने की वजह से agglutination या clumping होती है antigen and antibody की, and that can be seen visibly. ठीक है, और हम detect कर लेते हैं कि yes, this particular person has malaria. So this means that this rule of using monoclonal antibodies, because antibodies are so highly specific, it can be used to detect any pathogenic infection inside your body, infection caused by viruses or by bacterium or anything else. ठीक है? Now, problems which we face when we use monoclonal antibodies produced by mice or rabbits or laboratory animals. Of course, when we make antibodies, monoclonal antibodies by using mice or rabbits, there is a chance that if such antibodies are given to humans, they might trigger an immune response, क्योंकि आखिर आई तो वो किसी animal के body से है ना, उसके plasma cell से. So this problem has also now been overcome by humanizing MABs. इसके बारे में भी आपसे सवाल पूछा जा सकता है. Why is there a need for humanizing MABs? So you will say MABs should be humanized so as to minimize the chance of an allergic reaction developing in humans when they receive monoclonal antibodies made in the body of the mice and rabbits. तो हम उसको humanize करते हैं. How can we humanize it? Of course, gene technology से humanize होता है, and it can be done. आपको सिर्फ दो तरीके बताए हुए हैं यहाँ पे. We can alter the genes that code for the heavy and the light polypeptide chains of the antibodies so that they can code for human sequences. देखो, हम सबको पता है प्रोटीन सिंथेसिस कैसे होती है. DNA makes RNA, RNA makes proteins, and if I can change the DNA, then I can change the RNA also, and hence I can change the polypeptide altogether. तो हम क्या कर रहे हैं कि if we could through CRISPR technology, जो अभी आपने नहीं किया और आपसे कोई एक्सपेक्ट भी नहीं करता कि आपको ये सब कुछ आए, but at least आप मेंशन कर सकते हो कि by altering the genes that are actually coding for the polypeptide antibodies, we can alter the heavy and the light polypeptide chains. इस तरह से alter कर दें कि जब antibody बने तो वो human antibody से बहुत resemble करती हो. The nucleotide sequence, I mean, right? Another way by means of which we can humanize antibodies, we can change the type and the position of sugar groups that are attached to the heavy chains to the arrangement found in human bodies. When we were doing the structure of antibody, हमने यहाँ किया था antibody का structure. Let me go back and show you that slide. We had seen that every antibody has a chain of sugar molecules, this polysaccharide chain, that makes the antibody stick to the surface of the epithelium inside the human body. तो अगर हम इस polysaccharide chain को जो आपको नजर आ रही है, this one, if you could change this also, we can humanize the antibodies. ठीक है? तो हम ये भी कर सकते हैं. तो the examiner आपसे जब पूछेगा, आप इन दोनों options में से कोई भी लिख सकते हो. Some MABs have had significant success in treatments that involve modifying immune responses. Are you guys supposed to remember the name? ये इतने मुश्किल से नाम. No need to remember this. सबसे ज़्यादा जो question आता है exam में, it is asked about Herceptin. So and also some one another also MAB. तो हम थोड़ा सा इसके बारे में पढ़ लेते हैं. There is a monoclonal antibody which is known as Herceptin. ठीक है? It is used in the treatment of some breast cancers. It is a humanized mouse mab. What is its name? Its name is Herceptin. किस चीज के लिए यूज होती है? ब्रेस्ट कैंसर की ट्रीटमेंट के लिए यूज होती है. Let's see how it becomes effective. अच्छा, अब तक जो हमने सारे एग्जाम्पल्स किए थे ना मोनोक्लोनल एंटीबॉडीज के, दे वर मेंट टू डायग्नोज डिजीजेस, दे वर मेंट टू फाइंड आउट द ब्लड ग्रुप और डू द टिश्यू टाइपिंग. अब हम किस चीज की तरफ आ रहे हैं? ट्रीटमेंट की तरफ आ रहे हैं. सो गर्ल्स प्लीज रिमेंबर, एग्जाम में जब क्वेश्चन आएगा मैब्स पे, तो आपको बहुत गौर से पढ़ना है कि वो क्वेश्चन जरूर कुछ ना कुछ आपको एक बैकग्राउंड इंफॉर्मेशन देता है. यू हैव टू रीड दैट बैकग्राउंड इंफॉर्मेशन केयरफुली. क्या ये किसी डायग्नोसिस के बारे में बात हो रही है या ट्रीटमेंट के बारे में बात हो रही है? ट्रीटमेंट के बारे में बात हो रही है तो उसी लाइंस पे आप सोचो. So right now we are doing a discussion on the treatment of diseases using MAV, and the first example that we are doing is Herceptin, used for treating breast cancer. Let's see how it does. Herceptin, इसका नाम मैं नहीं ले रही, इतना लंबा सा नाम है, we can just say Herceptin. It binds to a receptor protein that is produced in abnormal quantities in the cell surface membranes of breast cancer. यानी कि breast cancer, if a woman is suffering from breast cancer, then she has many abnormal cells, the cancer cells, and there on the surface of those abnormal cancer cells there is a receptor protein. तो ये वाली Herceptin antibody क्या करेगी? जाके उस receptor protein से bind कर लेगी. ठीक है? अच्छा, the receptor protein is not unique to cancer cells, but cells between the receptor protein is not unique to cancer cells, but cells with between 10 and 100 times the usual number of these receptor molecules in their cell surface membranes can only be cancer cells. So Herceptin binds to these receptor proteins and marks them for destruction by the immune system. ठीक है, तो क्या कर रहा है Herceptin? Herceptin is a monoclonal antibody which to the receptor protein found on the surface of the abnormal cancer cells in women suffering from breast cancer. ये हमारे पास भी प्रोटीन्स होती हैं, लेकिन अगर ये प्रोटीन्स क्वांटिटी में, if they are more than the usual quantity, it shows the pathologist or the physician that this is not how a normal cell behaves, it is a cancerous cell. So now when the monoclonal antibodies will bind to these receptor proteins, तो क्या कर लिया उन्होंने? Monoclonal antibodies ने इन cancer cells को tag कर लिया. So when they have tagged these cancer cells, what has actually happened? Now these cancerous cells can be easily destroyed by the killer T cell. This is a very, very, I would say a very hardcore concept that you need to understand and you need to remember also, right? Can I take 5 more minutes, girls?
Sure.
Another kind of a monoclonal antibody which is used for melanoma. Melanoma is a skin cancer. What does this do? It works by activating the immune system, but in a different way to Herceptin. Ipilimumab binds to a protein produced by T cells, the role of which is to suppress the immune response. By blocking the activation of the protein, an immune response can be maintained against the cancer cells. People who have melanoma, उनके अंदर there's a protein made by T cells जो कि क्या कर रही है? It's suppressing the immune response. Only in those people who are suffering from melanoma or skin cancer. We don't want the T cells to make that protein which will suppress the immune response. So what does this ipilimumab do? It blocks the action of that protein, and in doing so, the immune response can be maintained against the cancer cells. The question is, are you guys supposed to remember each and every mechanism? I think you can just remember these 2 examples. ठीक है, बाकी आपको कोई जरूरी नहीं कि आप सबका याद रखें, but you can just read them. Infliximab is used to treat rheumatoid arthritis, which is an autoimmune disease. This condition involves a protein secreted by T lymphocytes that cause damage to the cartilage in the joint. Rheumatoid arthritis is a disease जिसमें the person develops pain in the joints, बहुत शदीद उसको पेन होता है, वो इसलिए हो रहा होता है कि इनके T lymphocytes जो है ना, they are making something. What are they doing? Let's go back and see. The T lymphocytes in such people who are suffering from rheumatoid arthritis, there is a protein secreted by T lymphocytes, and this protein causes damage to the cartilage. और इसकी वजह से they have pain and swollen joint. So in them also we can give infliximab because it will bind to this protein that is causing damage to the cartilage and hence blocking its action. ठीक है? Most of those treated for rheumatoid arthritis receive this therapy at monthly or 2 monthly intervals. So it is important that the MAB is humanized and does not itself trigger an immune response. Acha. Examiner can ask you this question also. And let me write this and I will share these slides with you. The question is, and with that, I'll just take 2 to 3 minutes and then I'll end. That explain why is it not necessary to humanize MABs used in diagnosis? And why is it necessary to humanize MABs used in the long-term treatment of diseases? Amna, can you answer this question? It's very logical.
It's because, like it was written in the book that our immune response, if we use an animal generated map that has not been genetically altered, our immune response might be triggered and it might cause problems. So if it's just for diagnosis, we'll be done with it quickly, and the maps won't have to stay in our body for long. But if it's a long-term treatment, then they have to stay for long and they need to suit our body.
Very good. See, diagnosis is a one-time procedure. Long-term treatment is not a one-time procedure. Okay? You might be needing to receive infliximab for a very long time. And let me tell you, girls, these monoclonal antibodies, जो है ना, ये बात करने में बहुत मजे की लग रही हैं. The treatment of monoclonal antibodies is very, very costly. It can cost in rupees, I can say 30 to 40 lakhs, right? Even more than that. And it's not for one time, it goes on sometimes for years. So it's a very costly treatment. It also has high risk side effects also. So that's why. And side effects में of course allergic reactions हैं, बहुत सारी type की चीजें हैं. Even humanize होके भी ज़ाहिर है, it's not the same antibody जो कि आपकी human body बनाती है. So the risk of infections and the risk of side effects will always be there. Okay, I end over here and I have completed this unit. Girls, start revising, not revising actually, yes, revising plus also doing past paper questions on monoclonal antibodies. Recent वाले past paper questions आप करो 2020, 2021, 2022. उसमें देखो आप कि किस तरह के questions आ रहे हैं